So it's a great pleasure to welcome Samir EL Andaloussi from the Karolinska Institute in Stockholm, who's been working on extracellular vesicles for quite a number of years now. I think it's beyond the 8 years. Samir, you'll tell us more detail. And you're also helpful in starting EVOX, and you're a consultant for EVOX, and I know your lab has been supportive in making EVOX successful. And you're going to be joined by André Görgens, who is working with you, and he was working with extracellular vesicles previously at Essen with Bernd Giebel and others on mesenchymal stem cell vesicles, I believe. So welcome, guys, and we look forward to your presentation.
Thanks, Jan. Thanks for the kind invitation.
Yeah, thanks.
So actually, this is my first time to do a web talk, so bear with me if things are too slow or whatnot. What we will talk about in general is the use. I mean, what we do in my lab a lot is trying to engineer EVs for various biomedical applications. And I will start off by just generally introducing our main topics, show two examples or two projects where we've used EV engineering either to address biodistribution with a high resolution in vivo or to use them as multimeric biologics. And by the end of my talk, André will then go over and more in greater detail, tell a little bit more about the analytics that we do, and in particular the single vesicle analytics that we do on various engineered EVs. So, as Jan pointed out, this is a disclaimer just that I'm a co-founder and shareholder in EVOX Therapeutics that develops engineered EVs for various diseases. Obviously, some of this work we've been doing on a collaborative basis at KI, and that is not the part that I will present today. So I think you all know what EVs are, so I'm jumping directly into the topic of my lab, and that is trying to develop EV therapeutics. And when we talk about EV therapeutics, you could broadly divide these into 4 different classes. You have native EVs. With native EVs, I mean EVs that are directly derived from cells that has an inherent biology on their own. Mesenchymal stem cell EVs being, I would say, the most exploited source of EVs for treatment of various inflammatory conditions, but also for tissue regeneration. Then we have the hybrid EVs that has emerged more and more in recent years, and that's basically as the name implies, a hybrid between a extracellular vesicle and any other chemical vector. Then we have the exogenously engineered EVs, and that's basically when we take a drug and put it into or onto an EV exogenously. And the last approach is what we refer to as endogenously engineered EVs, and that is where my lab has primarily interest in using. And I can say that largely what we are interested in is trying to deliver all sorts of macromolecular cargoes to tissues beyond liver. So trying to contrast to what you can achieve with lipid nanoparticles that are now being clinically used. So to generate engineered EVs, as I said, there are 2 strategies. To the left, you see that what we refer to as the endogenous engineering approach. This means that we transduce cells with an EV sorting protein fused to a protein of interest, then have that expressed in the cell. And then the EVs that are released, depending on how they have been engineered, can be either to display a targeting moiety or to load a protein on the inside of a membrane protein in a vesicle, to express a therapeutic protein on the surface. The pointer doesn't really work here. It's a bit annoying. Is it moving? Okay, it can be a therapeutic protein on the surface, and that is what I will tell you a little bit more about today. It could be display of RNAs, it could be display or loading of RNAs through the use of RNA binding proteins. On the right side, basically, you take instead EVs that has already been isolated, cells have not been engineered, and you put these molecules into the EVs. And that is then referred to as the exogenous loading approach. So the two stories I will tell you today is the first one will revolve around how we can use EV engineering to study biodistribution with high resolution in rodents. And the second part of my talk will be on the surface display of biotherapeutics for treatment of inflammation. So, and what is common for all the projects that are running in my lab, this is basically the workflow, the purification flow. So what we typically start with is we have exosomes, or we have cells, I should say, grown either in 2D cultures or in hollow fiber bioreactors or in batch suspension reactors, depending on what cell type we're dealing with. We're then typically concentrating the media that is then the conditioned media from these cells through tangential flow filtration, hollow fiber based, in order to concentrate the volumes to a manageable volume. Then, depending on how pure these EVs are, depending on what media they've been grown in, we use them straight away, or we go to the next step, which is size exclusion separation of EVs, where we then can separate the proteins in the secretome from the vesicles in the secretome. And once we have the EVs, we're ready to use them, or optionally then for exogenous loading, you go through an additional loading step of the isolated vesicles. And these are some of the EV analytics that we always use for our EVs and engineered EVs. Now this is a phenotyping of engineered exosomes that I will talk about in the end of my talk, but basically what this includes is nanotracking analysis, Western blot. We do bead-based flow cytometry to look at the vesicles. We run electron microscopy, and André will talk more in detail about the single vesicle flow cytometry by image-based flow cytometry that we do to look at individual vesicles. So this is just how this started. So in order to engineer an EV, or to bring a protein, in this case a protein, into EVs, a few years ago, what we set out to do was to take highly ranked proteins that we got out from proteomics, from, in this case, it's stated HEC293T cells. We've done this for at least 30 other different cell sources. The most common ones are actually also abundant across cell types. We then fuse these, in this case, to GFP. And as you can see on the right side, there is a number of different constructs and designs. We've then done single vesicle analytics on these EVs, either by regular confocal microscopy or by fluorescence correlation spectroscopy. And as you can see on the right panel, the conclusion of this study that we published last year in JEV is that transmembrane proteins tend to sort the GFPs better than the soluble fusions. And based off this work, we then wanted to take it one step further and see, can we use these engineered vesicles in any way to study their distribution in vivo? Because traditionally we've done lipophilic dye staining of vesicles, but for different reasons we are not very satisfied with that, because at some point in time it may be that we are no longer tracing the EVs but actually the lipophilic dye. So to avoid that, what we did is we replaced GFP for a range of different luciferase enzymes, and in initial pretest just checked, okay, if we engineer cells to express these and sort these into vesicles, what are the light emission that we can get out? And what you can see on the left side, the high bar there is when you have nanolook EVs, and that is also depicted on the right-hand side, where we've just taken a photo in the dark room with a mobile phone, and you can see that they light up blue. The bottom range, where you have the nanolook labeled, that is when you do a triton treatment and proteinase K. The middle row is if we only do proteinase K, and the upper row is the vesicles. This means that we are also sure that we're loading the cargo-based detection is on the inside of the vesicles, not on the outside, so we don't have the risk of getting shedding in plasma. We've then taken this system and applied it. We've used different cell sources, made them genetically stable for these nanolook or thermal luciferase construct, and then ran biodistribution in mice after different injection routes. What the left bar graph here depicts is an intravenous injection of cord blood-derived MSC EVs, and then we've taken out organs at 5, 15 minutes, 30 minutes, 60 minutes, 3 hours, 6 hours, and 24 hours, and looked at the signals in each tissue. And since we know how many RLUs we have per vesicle in different biofluids, we can then recalculate the number of EVs in each organ at any given time point. And what you then can see is that liver and spleen, no surprise really, are the highest organs, but those are also the organs where the signal decline fastest over time. To the right is what if we take out plasma at different time points, and you see how much is left of the injected EVs. And we have a little bit difficulty actually determining half-life because the tissue penetration of the vesicles is so fast, so it leaves blood, and we estimate that the half-life in this case is around 2 to 3 minutes. In order to be able to visualize biodistribution in real time, we have opted to, instead of using nanolook, we've also used the same genetic construct for spannings, but using thermal luciferase. And the beauty with thermal luciferase is that allows us, if we inject the substrate just before the EVs, to image exactly where the EVs go in real time. And as you can see, there is a very rapid uptake into lungs that then distribute to liver and spleen, very similar to what we saw with nanolook EVs. And in the bottom, you can see the dose that we used. In this case, it's HEC293 derived vesicles. So we know that how the distribution looked. The distribution is quite body-wide, but it is, as usual, with non-viral vectors, primarily liver, spleen, and lungs that take up EVs. Now, I will show you one slide, and I won't go into details since this is very early on in a study, but I will say that although the distribution between different vesicle sources from different cell types look very similar grossly, we have, by single-cell RNA sequencing, been able to see that actually, that if we look at 2 different exosome sources, in liver, for example, we see that this blue color is where the cell type where all exosomes in exo1 group clusters, whereas in the upper you see that this is the liver from exosome source 2. And the difference between those two is that with one cell type, we get EVs that accumulates in Kupffer cells, whereas the other cell type, it only accumulates in hepatocytes. And if we then take that through spleen and lung, we see very similar trends that whereas one cell type ends up in macrophages, we see the other cell type predominantly going to B cells, for example. So this is very exciting, and this is something that we will pursue further, because even this, if you look at global biodistribution, doesn't actually tell you what cells exactly in each tissue that takes these vesicles up. So this is one example of just how we use EV engineering to study clearance rates, distribution over time, etc., etc. The next part of my talk, which I hope that we have a few minutes for that, is how we can use EVs as basically carriers for biologics, to bring biologics in numbers in one package, so to speak. And this is actually a story that eventually has gone now into a manuscript stage. We started working on this actually 7 years ago. That's almost only a year after I entered the EV field. And it's based on the principle that we modify source cells to secrete EVs. And these EVs on the surface are bioengineered to have signal incompetent cytokine receptors. This means that they can avidly bind cytokines, but if they are delivered into a cell, they will not signal. And this is how an engineered EV with decoy receptors look like. I will not go again into details because we don't have time for that, but as you can see from this slide, this is just a snapshot of all the genetic constructs that we put into either HEC or MSC cells to find the best construct that can sort cytokine receptors to the surface of EVs, but also bind cytokines with high avidity. We started off, like most projects, with CD63. We have then moved down to more advanced engineering principles. In this case, it is for sequestering TNF-alpha receptor on the left side. On the right side, we have the GP130 or the IL-6 signal transducer, which is the co-receptor for the IL-6 receptor. And the idea with using this IL-6-ST, the signal incompetent, is that it can bind IL-6 receptor complex in blood and thereby block IL-6 trans signaling that is important for many autoimmune disease and chronic inflammation. So in this screen, so this is one example with a TNF-alpha receptor. We use a reporter model system where it's on the right side, where you have the TNF receptors are expressed on the cell surface, and inside we have a reporter system that measures NF-kappa B activation that drives luciferase expression. So if you add naked free TNF alpha, it will bind to the receptor and it will induce signaling. However, if our decoy EVs have a biology and does what it's supposed to do, binding TNF alpha, you block this TNFR signaling. And if you look at the bar graph, so we have, in this case, compared 4 different EV sources. So control EVs, they are non-engineered, they are in black. And as you can see, during a dose response, there is no real dose response because these EVs do not interfere with TNF-induced signaling. However, if we now then take one where we have taken the TNF alpha receptor, mutated in a matrix metalloprotease cleavage site that is outside in this genetic construct, and put an N-terminal syntenin sorting domain on these EVs, we start seeing that you get the dose-dependent reduction in TNF-induced signaling. And interestingly, if we now start introducing oligomerization domains, so we call this fold-on, to trimerize the receptors, which more mimics what happens actually on a cellular level, we see that we can robustly increase this potency in these dose response experiments. And depending on if we put it on the outside or the inside, it seems that putting them on the outside has a benefit. I didn't include a graph to have them both on the inside and outside, but it doesn't really provide any additional benefit. Moving on, so the same screen, but in this case now to look at IL-6 trans signaling, and you can see the designs on the upper part of this, this picture. Same principles, basically GP130 in this case instead, or the IL-6 ST without the signaling domain. A sorting domain, again an N-terminus syntenin that will help dragging these into EVs. An oligomerization domain, in this case, it's rather a leucine zipper because these receptors are meant to dimerize. And the reporter readout is on the right side. In this case, what we measure is STAT3 phosphorylation. IL-6 signaling will induce STAT3 phosphorylation. And in this case, STAT3 phosphorylation drives the expression of alkaline phosphate. So if you look at the bottom left, what you will see is that with control EVs, we have some effects, but they are not significant on IL-6 induced signaling. However, if we use our leucine zipper construct or the N-terminal syntenin, you see a beautiful dose-dependent reduction in IL-6 induced signaling. We've also played around replacing the N-terminal syntenin with a transferrin receptor N-terminus domain to see if that sorted better into EVs, or even including others like tetramerization domains. Most of these do not perform better. So in all subsequent work, it will be the dimerization domains with the N-terminal syntenin that I will show. So this was transient transfections, but you know that transient transfection can also be carryover from transfection reagent in different contexts. So we wanted to make stable cell lines. So this is work that was done in Oxford with Mariana Conchizao, where she generated 2 stable cell lines, HEC293T and MSC. And then we took EVs and did the very same dose response experiments. And as you can see, similar to the first one, is that the second generation, we call them second generation EVs, are better than the first generation, which are the simple transmembrane domain. And the same thing in both in MSCs and HEC. And when you look at the Western blots down, you also see that we get more of these receptors when they have the dimerization domains there into the vesicles. We proved the biological activity of these EVs beyond reporters by looking at the muscle differentiation in vitro first. So it's known that suppressing IL-6 signaling stimulates muscle growth or muscle differentiation. And what we can see here is that if we dose dependently add in EVs, you see that we get less phosphorylated STAT3, and that's also depicted in the bottom left graph when you have the decoy EVs. And if we look on the right side, what you also see is that treating these with the decoy EVs partially or mostly actually rescues the differentiation process that is blocked when we, in the absence of EVs, or when we treat with only wild type EVs. And these are MSC EVs. And this is also confirmed in vivo. So here Mariana and colleagues have injected the EVs and then imaged different muscle groups and see that they actually do go to some of the muscles. When we then look at phosphorylated STAT3 either in quadruplex or gastronemicus, this is in Duchenne muscular dystrophy mice, we also see a robust reduction in in STAT3 signaling. So it shows that these are biologically active in vitro, in muscle, and in an in vivo model. We next did a similar experiment, but now moving into inflammation models, where anti-IL-6 and anti-TNF drugs have been used for years. And what this graph shows to the left is that when we generate these stable cell lines, we also get beautiful dose-dependent reduction in TNF-induced signaling compared to using mesenchymal stem cell EVs only in the TNF reporter. When we compare this on the right-hand side upper graph, we see that actually the IC50s is about a log lower than with the clinically used etanercept receptor decoy. And the bottom left is showing one of many in vivo trials. This is an systemic inflammation model that is induced by LPS, where we've then either treated with control EVs, EVs engineered with a TNF alpha receptor, or etanercept. And as you can see, the decoy group performs better than the two others. Lastly, we have done a lot of work with the help of Roos van der Brugge in Ghent, running multiple sclerosis mice trials. We've also done some internally here. And I'm just showing you two different versions here. It's the IL-6, on the left-hand side, it's the IL-6 receptor decoys. And if you look at the clinical scores, a high clinical score means that they are doing really bad. When you're reaching these levels as the mock-treated group, they are basically hindline paralyzed. And we can see that we nicely, significantly rescues that by treating with decoy EVs. And very similar on the right-hand side, in this case, we've expressed an alpha body against, so it's an IL-23 receptor EVs, either pre-symptomatic or post-symptomatically, and then looked at the clinical score over time and at the end of the experiment. And you can also here see that the phenotype is rescued. And so this basically shows that these individual decoys do have inherent biology. We can suppress inflammation robustly in vivo. What we are now developing is what we call multimeric decoys. So the advantage with a vesicle is, of course, that you don't need to express only one receptor type on the surface. We can now start expressing multiple receptors. And this is just one example in vitro where we have co-expressed. So we've double engineered mesenchymal stem cells to display both the TNF signaling competent TNF alpha receptor and the IL-6. If we now look on the left-hand side, that is measuring IL-6 induced signaling. What you will see is that the TNFR EVs do not basically do anything in this, neither do unengineered EVs. But IL-6 engineered EVs, of course, do suppress the signaling, and so do the double decoys. We do the same experiment in the TNF alpha receptor reporter cells. We see that the control EVs and the IL-6 EVs, they don't do anything, whereas we see a nice dose-dependent reduction with the MSE, the TNF decoy EVs, and with the double decoy EVs. So we are now also have some data in colitis. I didn't have space to include that, but it really shows that we can now build in several receptors on these vesicles to get more robust suppression. And finally, I just want to give you one slide because it connects well to what the phenotyping work that Andrew will discuss after me, is also how we've started to engineer EV surfaces to display the antibody of interest. And we do this by genetically introducing a plasmid. In this case, I've written CD63. We have a range of other sorting domains. And then we have GFP, but in between we have what we call the Z domain. The Z domain is a Staph A FC receptor, or FC part that can bind an FC receptor. So basically what we do, we transfect these, we have EVs being secreted. These EVs will have CD63 on the surface, and they will have FC binding moieties on the outside and a GFP on the inside. And we've used this as a successful way to actually target EVs to certain receptors on tumor cells, but also targeting transferrin receptor. And Andrew will tell more about how we actually can use this as an analytical tool and also as a standard to look at labeling individual EVs. And with that, I would like to thank my team at Karolinska, which is now growing rapidly. Predominantly Dano, Joel, and Oscar for a lot of work on the decoys. Andre, who will talk after, on all the phenotyping, and of course the rest of the team. At the University of Oxford, I would like to thank Matthew, obviously, together with Mariana and Imre. and then all the folks at Evox, Nicole and Wolf for a lot of the fluorescence correlation spectroscopy work that we published in JEB last year, Ross for help with the EAE model, and Itchy and Fiona, my old postdoc, for some of the cloning work in EV phenotyping. So now I leave the word to you, Andrew.
Thank you very much, Samir. Yeah, I think we move right away to André, and then we go to questions afterwards. André, please.
Okay, thanks a lot. Hi, everybody. So I will just talk a little bit about why sensitive analytical methods are actually essential. And you have seen that Samir was mentioning the workflow, or showed the workflow that we use just as a schematic. I mean, this just shows that we have many decisions coming from engineering cells or selecting EV producer cells to the final EV product that we then want to explore in preclinical studies or in research projects. So I would just like to emphasize that there are a lot of decisions through that workflow. So which cell type would we use? How do we best engineer the cells to produce certain EVs? Which constructs are we using? How do we scale the culture up to produce the conditioned medium, and which isolation method do we use to concentrate and isolate EVs, and how do we formulate and store EVs at the end? So it's just incredibly helpful to have appropriate and sensitive analytical methods for all these decisions that you make, because otherwise you make less informed decisions. And of course we use different methods to study function, to compare constructs, to measure size and concentration of particles, to do proof of concept electron microscopy. And what has been especially helpful for basically all projects at the end is flow cytometry. And I would just like to quickly mention or summarize and give some examples of two methods we are using in our lab regularly and combine. So the first one is a multiplex speed-based flow cytometry assay. It's a multiplex assay that actually enables you to, in a semi-quantitative way, assess surface signatures of proteins on the EV surface. So the workflow in brief is that you have 39 different antibody capture beads, and each of those beads is labeled with a different capture antibody. Then you incubate those beads with your EVs. The sample doesn't have to be purified, it can be done with conditioned medium directly. And then you incubate that. EVs with a certain protein on their surface will bind to respective beads, and then you stain with an antibody of your choice. By default, we use a pan tetraspanin cocktail to stain most EVs. And then you get nice signals, as you can see below, compared to a buffer control, which just does the exact same staining, but without EVs. You see for HEC conditioned medium, you get some signals. If you then gate on the brighter bead populations that are shifting up in this multiplex assay, you can see, and you can identify those, of course, and can see those are like the usual candidates plus an integrin. And the data type or the data quality you get out from such one tube, roughly like 30 microliter of conditioned medium. It's a surface profile of 39 or 37 different markers plus 2 controls. You see the tetraspanins on the top that are nicely being picked up in HEC EVs or on HEC EVs. You see also some integrins coming up and some other markers. And this is an assay we have looked in at much higher detail to make sure it's reproducible and we can use it in different contexts. An interesting figure is probably this dilution series, where we just looked at how much, how the sample input actually affects results. And you can see if we put an equivalent of 5e6 particles per assay in that assay, you can already pick up the tetraspanins, CD9, 63, and 81 up here, but you don't see basically anything else above background levels. And when you increase the dose then, you see that other markers are coming up, and like between 5e7 and 1e8, you see that, for example, CD29 or CD41 or CD49e are coming up higher. And this is, of course, showing the specificity if the signal increases over time, but it also shows that it's relatively sensitive. And of course, it also shows that in any other method, as well as this method, we have to be careful to interpret if something is negative or is not detected we can conclude it's negative or it's not expressed on those EVs. So if you get no band in your Western blot, you can't conclude the protein is not on your EVs. And if you get a negative signal in this assay as well, you can conclude that. You have to be sure or be aware that there's some limit of detection and that you might need more input. So just quickly mentioning which sample types can be analyzed. It's been very helpful to evaluate isolation protocols. So you can compare, for example, in this case, conditioned medium with EVs isolated by different methods. As you can see here, ultracentrifugation via differential ultracentrifugation versus ultrafiltration. And you see that we see some differences. But it's here more like a proof of concept that you can actually study in process while isolating EVs all the different fractions. You can also study or use it to compare cell lines, and I'm not going in the detail due to time, but you can see that HEC EVs and MSC EVs have different profiles. For example, we don't see CD9 being picked up by those MSC EVs, and you can also vary the detection antibody to get more insight which markers are actually co-expressed with that assay. Since we are interested in therapy, of course, we also tried mice that were injected with human EVs, and we collected the blood, made the plasma, and then checked if we can detect those EVs still. And you can see if we have an untreated mouse, you don't get any signal. And if you look at the plasma from a treated mouse, after 1 minute, you get quite nice signals. Again, no CD9. The MSC EV phenotype is being picked up. And as Samir mentioned, the EVs are cleared very fast. And you see basically not much signal left after 30 minutes already. Now, just quickly mentioning or summarizing the other method that is called imaging flow cytometry. It's a single EV imaging method. So you get information about each EV, and you can do that with multiple parameters. We used GFP tagged EVs initially to learn more about how to best use that method to refine protocols and etc. And you can see that in those GFP tagged EVs are being picked up in the scatter low, so they don't scatter much light, but they are nicely gateable as GFP positive. And you can see the image data for each spot. We also did much more work to make sure we are really imaging single EVs and quantify those appropriately. Just one example I would like to show here is, I think, a big advantage of that method, that you can also, again, analyze all fractions. So if you start with complete THP1 CD63 cell culture medium, you see you can detect, if you look at the images, which is one of the big advantages of the method, you can see that you can detect the cells together with bigger fragments that are GFP labeled, together with different smaller EVs, smaller particles that are fluorescent at the same time. So it has quite a nice range in that sense. And what you can then do, if you go through the fractions during an EV isolation, if you do a 2000 G centrifugation, you already get rid of the cells and bigger fragments in R2 and R1 here. If you do the 10,000 G centrifugation, you get this much more cleaned up and enrichment in the scatter low area. And if you then do an 0.2 micrometer filtration, you basically end up with this population shown here in R5, which is GFP positive smaller EVs that are scatter low. And if you do an ultracentrifugation, you basically see that population again. So that shows that you can use that method again to evaluate isolation protocols. And it also gives us more confidence that what we look at are actually very small EVs in this scatter low region. Of course, we don't want always to analyze labeled EVs with GFP in particular, but this has been also a very helpful tool to optimize antibody staining techniques. So the long-term goal would be to use several antibodies to stain EV subpopulations or of course qualify different preparations. This is just showing that if you have unstained GFP EVs and versus CD63PE stained GFP EVs, you get a nice separation or a nice contrast of that population. You have to be careful about how to do that exactly, and we currently use that system, or we have used it before already, to titrate antibodies to optimize the system and so on. So it's definitely going towards a step where you can use antibodies and combine them to learn more about EV subsets. And as Samir mentioned, it's also quite interesting to use those Fc binding EVs in that context. Of course, in many cases, we just use flow cytometry to evaluate the EV constructs that we make, to make sure that if we engineer a protein to be on the surface or inside, that it's actually expressed, and compare different constructs and make some controls or control for expression in general. So these Fc binding constructs can of course be used with that method as well. So if you just compare on the top row, here you can see control EVs treated with increased amounts of antibody, which is an isotype control in that sense, so we are pretty sure it's an Fc binding. And in the lower section, you see that you have dose-dependent binding of this isotype control in APC with only to those Fc binding EVs. And here you can see some more steps where we did some titration and can study nicely in this context the binding and also qualify the constructs as well in parallel. So currently, just on a high level summarizing, we use both methods quite regularly for different questions. And it's basically the bead-based flow cytometry gives us quite a fast snapshot of which proteins are expressed. We use it for screening, comparison of many cell lines, and the image flow part then gives us detailed information about all the rest. And I would just mention very quickly that we also started, since there's apparently quite some interest in EV flow cytometry and also quite some need for support and education in that sense. Together with the flow cytometry working group from ISEF, ISAC, and ISTH have started an online educational series. Actually, yesterday I gave the first talk, and it had already 330 attendees. So that was quite surprising. So if you're interested, maybe feel free to join that as well. It's quite nice to see all these online talks coming up, and it's quite a nice opportunity for everybody, I guess. Yes, I'm also thanking everybody who has contributed to the project, of course, especially mentioning that the image flow part was started in Bernd Giewitz's lab in Germany, in Essen. Thanks.
Great. Thank you very much, André and Samir. That was very interesting, and hopefully we can also put this up on the internet. It's going to be a very popular video, I'm sure. So this is up for discussion, and we have a series of questions on the chat, but I think Carolina, will you help unmute persons that like
yes,
Will?
so now everyone can unmute themselves. So I think the first question is from Will. Will, you ready to ask question? Please go ahead.
It was just a very basic question, but I've seen it a few times on some flow analysis. André, it was just on the gating for single beads only. There was sort of like a second population sort of above and to the right. Was that just sort of doublets of beads going through the flow cytometer?
Yes, exactly. So that's just doublet. So you use, sorry, the scatter properties of the beads and plot them against each other, different parameters, and basically you get double the signal for doublets on a log scale that is then right next to that population. So we make sure we only gate on single beads.
Okay, thank you very much.
Okay. Donovan, please go ahead.
Hi, thank you, André and Samir, for a great talk. For the first half of the presentation, I'm curious about those mouse studies because they look pretty amazing. My question is, are you able to produce enough EVs at an affordable price? And in the future, when you are looking at potential human trials, are you able to produce them in a sufficient way?
So for preclinical work, for the work we're doing in my lab, we can produce them at sufficient quantities. It is obviously comes with a cost. It takes a lot of space. If you do 2D cultures, you need a lot of plates. Some of these experiments we're talking, if we do a big animal trial back in the days when we started this decoy work, for example, we could have 515 centimeter dishes growing. Of course, for a lot of this, we moved to do it using bioreactors for some of the work with the MSCs. Then, of course, that simplifies life because you get a lot of EVs out from these 3D kind of structures. what I would say also is the fact that we got rid of the ultracentrifugation for the in vivo work many years ago also made it a little bit easier to handle, to run the TFF system. But and clinically, I mean, I would never do that, obviously internally, but we think that some of these workflows, this is similar to what I think the companies that are now trying to develop EV therapeutics, that the same, very similar workflows as they are using. I think, but you know, really the limitation is how can you grow cells at scale? Obviously, I mean, regular 2D cultures is not going to be beyond preclinical work.
Thank you.
The next question was Umaima.
Hi, so I'm just curious, what is the difference between the EXO1 and EXO2 that they used? Because there was, they targeted different cells, that's why.
Yeah, it's two different cell sources.
Ah, okay.
Yeah. They're derived from two different cell sources.
Can you disclose which ones?
Preferably not now. It's more, I think it's just more relevant to say that there are differences, but I would prefer not to, if that's okay. I think actually, to be honest, everything that I presented is unpublished. So I think I've been quite fair with trying to divide, share as much as I possibly can.
That's good. Yeah, I mean, you know, we're curious, and especially what are the surface molecules that distinguish kind of selective uptake and stuff like that?
And these are the things, exactly the things that, you know, Andrew will help me figure out with the analytics at the later stage.
It ain't easy, right? It's not that easy.
No, of course not. And you have another layer, which I didn't discuss. Of course, you have protein adsorption. You know, it's not just how an EV looks like. An EV is an EV, but an EV in blood is not the same as an isolated sample in vitro. And that we know, and it clearly behaves differently.
Yeah. So, Sharif, Ibrahim?
Yes. Thank you for this very nice talk, informing. And so my question is about the decoy proteins, because some people already started trying to use these decoy proteins, but they use them as soluble. I didn't see anyone who tried to put them on the EVs before. So this is a very nice idea, but I was wondering if you already tried to compare when you use just direct soluble ones versus EVs.
That's what I showed. That's what I showed. So etanercept is exactly that. So that is a soluble receptor.
This was one in one of the figures that showed more effect when it was okay. I missed it somewhere. Okay, thank you.
And I mean, honestly, when I started this many years ago, I was of the naive impression that EVs are awesome because they circulate forever. I still think EVs are awesome, but they do not circulate forever. But what they may do is entering tissues, and that we know by imaging that they do, and it may be the reason they work better than the regular biologics. I mean, with a few experiments we've done, can be that it's protecting locally in the tissues rather than just circulating in the blood.
Yes, okay, thank you.
Elisa Lazaro Ibanez.
Yeah, thanks, Jan. A very nice talk. Thank you very much. My question is about the in vivo biodistribution data. And I was wondering if you could elaborate a little bit about the fact that nanoluxi 63 EVs accumulate in the lungs. Is it something related with, you know, aggregation when these EVs reach the bloodstream? I assume that you are IV injecting these particles, or is it about a change in the tropism of these engineered EVs? Because when your group and others have done, you know, just fluorescent label with near infrared dyes, or others have reported data on radio-labeled EVs. Indeed, we see mostly accumulation of these particles in liver, followed by spleen and kidneys. Yeah, so is this different biodistribution a result of, you know, the whole process of engineering?
So my answer would probably be no. It's a little bit higher in the Thermalock experiments. That's because we have used a higher dose. So there may be a degree of lung capture because of size. generally speaking, lungs or liver and spleen are always highest in our hands. And that comes as no surprise. I think this is any nanoparticulate that you get in lung, and when you do the real-time imaging, is that is your first capillary bed that you hit. It's not liver.
Yeah.
I mean, this is your lungs is the first that you face, no matter how you do an intravenous injection. So naturally, you will have some. Then the tropism question is, we do not see any major differences between EV sources. That doesn't mean that there is no tropism difference. That just means that the distribution looks similar. So I don't think it has to do with engineering, because it's the inside also that is modified.
Have you tried to do
that slide? But we have a slide. I didn't, because we didn't have time. We've done a very interesting and informative experiment where we used nanolook labeled EVs, and then IR dye labeled them, and then we've monitored at the same time points nanolook signal and IR dye signal. And that's why I'm saying that when you put an IR dye there, you do have a little bit of impact on the EVs, because the signal moves.
Okay, but that's a nice experiment. Yeah, thank you.
I'm happy to share that. And it's actually, we are resubmitting that paper hopefully in a couple of weeks, and it's included there.
Okay.
Benedetta Busalati.
Yeah, so thank you. Really nice presentation. So I got quite excited by your data on the blood urine that you could, on the blood vesicle that you could detect with the beads. So the injected vesicle that you were detecting. So did you isolate the vesicle from the blood? Was it a direct analysis of plasma or serum?
Yeah, no, I didn't mention the details, sorry, but we just generated plasma, and without further dilution, we put 30 microliters of plasma in that assay and got those results. So if you would isolate, you would get higher signals, I guess. But it was for that stage, it was a proof of concept. Generally, we are working to improve this a bit more and use it for comparison. I mean, the beauty of this is that you get more information about subsets potentially. And we also use the imaging flow for using human-specific antibodies in mice currently to study more like how many EVs are actually where, and ideally which ones, but that takes quite some time. It's just beginning, but yeah.
So that's going to be very useful. So my question was also, did you check also on urine or other fluids?
No, we didn't yet. I jokingly asked a colleague if we could get tears maybe, or saliva. That would also be interesting. No, we didn't check urine, but I guess you would find a bit.
We have checked all body fluids ever with the bead-based system.
Yes, was just about to mention. So we checked out of interest. I mean, we have compared like roughly 50 different human cell lines, all the different tissues with that assay, and also 10 body fluids, which each like 5 donors. To see what kind of phenotypes we get there, just out of interest, and of course to understand EV heterogeneity a bit more, and that gives quite nice results. So if you use the equivalent of 10 microliter of human urine, you get
we're starting doing that too in the lab, but my idea was really related to the fact of detecting human vesicle in mice. That seems quite sensitive because
of course that
shouldn't be too many. Okay,
it's very sensitive. And one thing is that, André, I mean, I don't know if you emphasize it. I mean, when you run just on our tissue samples or plasma, if you run a Western blot, you guys know how much EVs you need to put in those wells to actually detect anything. With this bead-based system, you're down 3 logs at least, and you properly detect the signal. And the advantage, another advantage is that you do not, so if you look at plasma and you get tetraspandin signal, doesn't mean that that's vesicles. It can be tetraspandin aggregates just going in blood, and we see that. But if you do the bead-based system, you're never going to detect them. So that is why it's so good to run both.
Yeah.
Thank you.
Okay.
We move on. So Joel De Beer has a broken microphone, and he's asking about the Duchenne model. How those EVs were targeted to muscle, maybe heart muscle or a skeletal muscle, or were they unmodified?
So the answer is they were unmodified, and if you include liver, lung, and spleen in those pictures, now, I mean, they were imaged separately. Of course, this is the minute signal of the total injected dose that ends up in muscles. But it's still very interesting to see that you do have the STAT3 inhibition still, although the effective doses are quite low. Muscle is notoriously, I would say, the tissue types where independently of EV source, where we have, it's a little bit more tricky.
It has to go through the endothelium, and then it has to be taken up by the skeletal muscle cells too. And they're not that active in their membranes, I guess, as macrophages or Kupffer cells and other things are. So
and I think this fenestration issue is really key when we talk about drug delivery in general, not just with EVs. It's really understanding that the endothelium lining is differentially regulated in different organs.
So Aaron, Scott, please.
Hello, thank you for a really interesting talk. So my question's for Andre, and I just wondered, I quite like the imaging flow cytometry system, but my understanding is that it's not possible with the technology to sort EVs at this point. I just wondered if you had any feelings for why this is and when this might be possible. I guess you don't really work for Amnis or anything like that, but.
No, I don't work for them. I ask them regularly if they can make that instrument, of course. But I mean, this will take much more time. I'm not sure if it will ever be used a lot, because you will dilute. It will take forever. If you sort single particles and you need like 1 × 10⁻10 or 11, that will probably take forever. So there are some papers out. You can check the lab of Jennifer Jones at NIH and some others, who have used an Astrios sorter to sort EV subfractions, but and they are sorted enough to make a Western blot, so it's conceptually possible, but it will dilute your sample a lot, because every drop is quite huge compared to the EV, and it will ultimately, not be applicable for like broader studies, I think, at least at the moment. Of course, it would be extremely helpful if you can sort subsets at a more efficient way, but it's maybe it works eventually. I guess for now, if we want to isolate subsets, the strategy is to find surface markers that are specific for those and then pull them down somehow and then analyze them separately.
So Pia Siljander, please.
Great talk, Andre and Samir. Really enjoyed it. I was just wondering if you had any thoughts about how the MHC1 complex will affect your EVs, or is the uptake so fast that it's irrelevant for the immune system? Thanks.
So, not entirely sure, but I can say I couldn't show data, obviously, but we've done dose escalation studies with Evox, so with the company that I'm working with, in non-human primates. Actually, pretty extensive dose escalations. There doesn't seem to be any immune responses associated of putting engineered human EVs into a non-human primate. That does not mean that, I mean, I can't really answer the MHC1 part.
Amazing talk, thank you very much. I just have a quick question. I don't know if you actually covered that in the beginning of the talk, but when you engineer your cells to target EVs to carry these receptors, would it be possible to target specific subsets of EVs like microvesicles or exosomes?
Yeah, it's a good question. I mean, first of all, I admittedly, in my interest is, I mean, I see them as vesicles. I do appreciate, I mean, the importance and the nature that there are different vesicles. I think there are two answers. We potentially could. I think that we have some single vesicle analytics suggesting that we know roughly which markers goes on a little bit larger vesicles and which goes maybe on the smaller vesicles. And we could, of course, use that, but we haven't at all looked at it. We have done some attempts trying to isolate by size, larger compared to smaller in the decoy context, because of course, from a decoy perspective, I mean, we always do 0.22 micron filtration, but that is for sterility issues. You can't go and inject a non-human primate if you don't remove that. It's very hard. But that is the sole reason. Actually, from a decoy perspective, I would imagine that taking larger vesicles too could have a beneficial effect, but we haven't been able to do that.
A follow up on that, because if you look at some of the literature of loading with engineering, Chang Lu, for example, claimed that ARRDC1 could be used for engineering molecules into the microvesicle. So would you comment on that, please?
Yeah, we haven't done the experiments. I know that they claim that, but we haven't We are about, so in 3 months I can comment on it, because that will be one of the constructs. So what we're currently doing, I can say, is we've ordered 230 genetic fusion constructs to systematically do a massive screen to actually understand, of all those top 200 proteins that we get across 60 proteomic data sets, what does sort best into vesicles. And ARDC1 will be one of them for comparison.
Thank you. Andrea Silva.
Hi, thank you for the talk. My question is to André. How do you guarantee that you are analyzing single vesicles in imaging flow cytometry and not, for instance, agglomerates?
Yeah, that's a good question, an important thing. To give a brief answer, we do a dilution series. I mean, we do two things. We do a dilution series and see if the concentration of EVs we measure are reduced as we dilute the sample. And it's important that at the same time, the brightness of the single particles we detect are staying constant. That's what we have shown. And on the other hand, we have the imaging data on top, so we can see how many spots we have per image, and we have applied an algorithm that actually distinguishes between the different spots. I couldn't show that, but it's included in that paper.
So thank you very much, guys. A big applause to you. It was a great fun 1 hour discussion here. So that was amazing. I don't think we need to edit very much. We can just put this on the YouTube channel quite soon. So Aaron Scott is helping us with that. So I hand over to Carolina to wrap up the session.
Yeah, thank you very much, André and Samir. It was wonderful and a lot of new ideas and exciting.